Okay. Okay.
That's true. We have to edit it down anyway, but yeah.
Okay. So yeah, just FYI, we are recording this because a lot of people that can't make it do visit our website and want to see the presentations through our website. So just FYI, let us know if you have any concerns with that. So welcome to the October 24th Science Work Group, everybody. I think we're just going to do a round of introductions. So just say your name, your affiliation, and then if you have an update on a project that folks would be interested in, let us. Do you want to go ahead and just do that while you're doing the introduction? So just to kind of keep time, we have a lot of presentations. And we usually almost go over with this meeting. So how about I'll kick it off. My name is Catherine Corbett. I'm with the Lower Columbia Estuary Partnership. And I'm just going to go down the list of names. So Andy, you're next.
I am Andy, or Andrea, and I am a research scientist on the monitoring team with the Lower Columbia Estuary Partnership. Hello.
Great. Thanks, Andy. Ann Creason.
Hi, everyone. Ann Creason, Bonneville Power Administration.
Jim Brick.
Morning, Jim Brick, Oregon Department of Fish and Wildlife, here in Clackamas.
Thanks, Jim. I think I might have skipped over Amelia Johnson. We're just doing introductions, Amelia.
Hey, good morning, everyone. Amelia Johnson, Lower Columbia Fish Recovery Board.
Thank you. Karen Adams. I might have skipped over you as well.
Oh, that's okay. I just signed on. Karen Adams with Washington Department of Fish and Wildlife.
Hi, Karen. And Curtis Roegner. You're gonna have to unmute yourself.
Yes, I'm Curtis Roegner with NOAA Fisheries, and I'm here with Susan Hinton and Jeff Grote.
Thanks. Derek.
Hey, I'm Derek from the Lower Columbia Estuary Partnership. Yeah.
Thanks, Derek. Elaine.
Good morning, everybody. Elaine Placido, Lower Columbia Estuary Partnership.
Erica.
Hi, everyone. I'm Erica Keeley. I'm with the Lower Columbia Estuary Partnership on the contracting side.
Thanks, Erica. Ken Fetcho.
Good morning, everybody. Ken Fetcho with the Oregon Watershed Enhancement Board.
Great, thanks. Ian, Mr. Wizard.
I'm Ian. I'm with the Lower Columbia Estuary Partnership.
And Jasmine.
Jasmine, the Public and Legislative Affairs Specialist at the Estuary Partnership, and according to wizardingworld.com, I am in Hufflepuff. I'm a Hufflepuff person. Just took the quiz.
What was that website again?
Wizardingworld.com.
Okay. Hey, Jason Toft.
Hi, good morning. I'm Jason Toft at UW in the School of Aquatic and Fishery Sciences, and our lab is the Wetland Ecosystem Team.
Thanks, Jason. Jeff and Susan, I think you were introduced with Curtis. Let me know if that is incorrect.
Yep.
Perfect. Okay. Jenny Dezo.
Hey, good morning, everybody. Jenny Dezo with the Estuary Partnership. And I'll share a brief update. The Multnomah Channel Marsh project that many of you have worked on, and I've worked on for many years, is in 60% design. So that's exciting.
That is exciting. Thanks, Jenny. Joe Needoba.
Good morning, Joe Needoba. I'm in the OHSU PSU School of Public Health.
Thanks, Joe. Kerry?
Hi, everyone. I'm Kerry, Accola, research scientist at the University of Washington. Is my sound coming through? I'm struggling on the audio part. It's coming through good? Okay, thanks.
Thanks. And the other Kerry. Sorry, Kerry.
Hi, I'm Kerry Dubler with the Estuary Partnership.
Thank you. Kirk Kruger.
I'm Kirk Kruger, Washington Department of Fish and Wildlife, and I'm one of the expert lead.
Great, thanks. Did I miss anybody? Oh, Sneha. What the heck? How did I miss you? Sorry.
Hi, everyone. Sneha from the Lower Columbia Estuary Partnership.
I'm missing a few people. Lynn Krasno, you're also missing you. And Sarah.
I'm just a miss. Lynn Krasno, NOAA Fisheries, and work on the SERP, the estuary program for the mitigating for the hydro system.
Sarah, I skipped you too. I don't know how. I think there was a button at the bottom that said more, and I missed that.
Sarah Kidd, I'm senior scientist with the Estuary Partnership, and I'm Gryffindor.
You're Gryffindor. Nice. Tawnya. Tawnya, I thought I saw you for. Yeah, there you are.
I'm like Joe. I'm in the OHS. Oh, are you not seeing me? Can you hear me? OHSU PSU School of Public Health.
Alex Uber.
Hey, good to see everybody. Alex Uber, Washington Department of Fish and Wildlife. Thanks for recording this. I'm going to probably have to step out shortly, but that's awesome that you're recording it.
Yeah, great. Thanks, Alex. Now am I still missing folks? Cool. I am gonna try to get out. Okay. So I think let's just get started. So the meeting is focused today on the ecosystem monitoring program and updates that we do annually. Other thing that I wanted to cover really quickly is the science work group. If anybody has topics for future meetings, please let me know. We used to meet like almost every other month, and we haven't been meeting in a little bit, mostly because I'm failing to come up with new topics that we haven't covered in the past. But we can get started in doing that if folks have some topics that they'd like to present on. So let me know. And you can do so via email later on. Okay, so any other updates from any of the folks on the call and they just didn't cover it during your introductions? And then Amy Kosorik, I think you just joined us. We just got done with introductions. Do you want to introduce yourself really quickly?
Sure. Yeah, sorry, I'm a few minutes late this morning. I'm Amy Kosurik. I'm a biologist with the NOAA Fisheries Washington Coast Lower Columbia branch located in Lacey. And this is Bob, my cat. I work on recovery of our threatened and endangered species and do Endangered Species Act consultations for the most part.
Great. Thanks, Amy. So Sarah and Ian, are you ready to kick us off? You have the first presentation, which is the overview of the ecosystem monitoring program and introduction to Tableau for data sharing and synthesis. So
Yeah, can everyone see my slide? My opening slide. So
Yep.
So first of all, I thank you everyone for joining us today. Really appreciate all of our researchers joining us today, as well as everyone who's interested, who's joined. And of course, we're excited to be recording this so we can share it with folks who weren't able to, carve so many hours out of their day today. Also, you'll probably notice that my team and I for this presentation are wearing our best wizard costumes. We just wanted to have a little bit of fun with it. We don't typically run a lot of workshops, and so if having the opportunity to have a little bit of fun today, we thought we would. I know we all sit through a lot of meetings and there's not a lot of magic sometimes. So today we're just going to try to integrate a little bit of magic into our science work group and spice it up a little bit. And yeah, anyway, without further ado, I hope that you can still take all of us very seriously with our wonderful costumes on, but also it keeps everybody awake. So first of all, I just want to say that I have the honor to introduce the Ecosystem Monitoring Program and the Science Work Group today, which is featuring all of the amazing researchers and the work that they're doing under this program. And some of the stuff I'm going to say this morning is super repetitive, and we say it every year. Before last few years, I've been saying it. Before that, Catherine was saying it. And honestly, I think it's such an honor and a privilege. And it's an honor and a privilege because this program has been collecting data in the estuary in one form or another for 18 years. This is a huge accomplishment, and I am excited to talk about it and provide an overview. And some of you may have heard it before. Some of you may have forgotten since last year. So let's just jump in. All right, if I can get my slides to advance. All right, so like I said, I'm going to introduce the Ecosystem Monitoring Program, the Action Effectiveness Monitoring Program. I'm going to briefly talk about the data that we collect in these programs. We're going to talk about the 2023 report, all of its different components. We have some new things happening this year. Last year, we talked a lot about how we're transitioning the workflow into a Tableau format, and so we're going to touch on that. And then I'm going to let the researchers take it away with their personal updates on the research they're doing and the progress. So when we think about the Ecosystem Monitoring Program and the Action Effectiveness Monitoring Program, what are these programs and what are their overall goals and objectives, like a really high level? So the Ecosystem Monitoring Program, its aim is to look at stats and trends of the ecosystem conditions in the estuary over time. And so we have these static reference sites, so ideal conditions through the reaches, that we're collecting a core series of metrics across monthly, annually, and this is a really consistent and a very large data set. And then to complement this, or to really feed, I really don't know why. Okay, I'm going to take a break to stop advancing my slides because I don't know why it is set up that way, but it was. Hold on. Okay, sorry about that. Let's get back into it. All right, because I don't necessarily want to go the pace that I went earlier when I was practicing. So the Action Effectiveness Monitoring Program is where we're collecting a similar set of metrics across our restoration projects. Now, these two programs work together. They work together because we take the restoration project data and we compare it to those reference sites, and it provides us a really good idea of what we should be expecting across these restoration projects in space and time, because as you know, climate variability occurs in the estuary and it's always shifting and changing. So without this long-term stats and trends data, we wouldn't be able to understand what's happening across our restoration projects. Again, these two also work together to inform restoration actions across the estuary. So overall, the overarching goal is just to understand the estuary, understand these ecosystem components, and do a better job of restoration and conservation moving forward. All right, so when we're talking about the different research components in these programs, what are we specifically referring to? Well, in the Ecosystem Monitoring Program, we had many of the researchers introduce themselves just a moment ago. We are looking at Mainstem, abiotic site conditions, so water quality across sites. That's mainly done by OHSU. Then we have habitat structure, hydrology, soil, sediment accretion and erosion, biomass, and detritus conditions. That is done in-house by the Estuary Partnership Group. Food web dynamics, really diving into what the salmon are eating and what those prey food items are also eating. That's going to be OHSU. Again, looking at those components, really critical work. And then, of course, we can't talk about salmon habitat without thinking about both the macroinvertebrates, which are studied by the University of Washington, and of course the fish themselves, which that research is led by NOAA. So all of these researchers are going to discuss this work that they're doing today in more detail, and so I'm not going to go too deep into this anymore. I also want to mention that we do get a lot of field support for this work by CREST. So April Silva and Ryan Elsmar are not giving a talk today, but they provide a lot of field support both for the EMP program and the Action Effectiveness Monitoring Program. We're really grateful for that work that they do. All right, so for the action effectiveness monitoring program. Now, today's talks are all really focused on the EMP program, but I really feel like you can't talk about these programs in a silo. So I always want to mention that we are collecting a similar suite of metrics across the action effectiveness monitoring program so that we can essentially merge those data together and answer larger questions about the effectiveness of restoration for recovering salmon in the estuary. Now, the history of this program, it goes back all the way to 2005, and it has changed in its iterations and the sites that have been involved in the program. But our core sites that we are going to focus on today have been, data have been collected on all of them since 2012, and some of them much longer. And those are the Ilwaco Slough at the mouth of the Columbia. So if I use my pointer here, I can point this out. So here we go. And everyone's probably going to show a similar slide where they talk about the sites that are collecting data across. So you have Ilwaco Slough at the mouth of the Columbia. You move upriver, you have Welch Island, you have White's Island. These are these essentially orange X's on the map. We've also included some of the action effectiveness monitoring sites on this map just for reference. Those are in blue. But we're not going to get into that too detailed. So then as you go up the river, you have Cunningham Lake and Campbell Slough and then Franz Lake. So the 5 core sites are Ilwaco, White's, Welch, Campbell, and Franz Lake. All of the researchers are collecting data or analyzing data across those 5 sites. Now, additionally, we're collecting data, and you can kind of see in this bubble up here, Cunningham Lake is an additional habitat site. And that is because in this reach where Campbell and Cunningham exist, there is a lot of grazing. Grazing really increases the variability of conditions that we see across sites. So we need more data to kind of understand what's going on with that influence of grazing that is happening across the reference and the action effectiveness monitoring sites. And then a handful of years ago, we added Steamboat Slough, which is an action effectiveness site. And we added that to our biomass study so that we could track biomass accumulation and detrital flux, not only on these EMP sites, but also at a restoration site. All right, so that is the overall sort of reach of the study or of all these researchers' focus. And let me go to the next slide here. Now, as you can imagine, I just gave a giant overview of the program. And this program collects a lot of data. There's just tons and tons of data across both the EMP program and the action effectiveness monitoring program. And for years and years and years, we roll these data into giant 300-page plus reports. Last year, we gave a really long and detailed talk about how we needed to evolve both our data management as well as our reporting to be more relevant to both our researchers and to folks who are interested in our program and our data. And so we've been really working this year, really one-on-one with all of our research groups to start restructuring or re-evolving their data database system so that we could flow that data into Tableau. And this is, as you can tell, if you look at any of these numbers, we have millions of data points, various different types of data, and we're continuing to evolve both how we integrate Tableau into this work and how it can be utilized to help our researchers do more cross-pollination with each other and do more synthesis work. And so when I talk about the Tableau transition, I want to just really feature the new report. So last year we talked a lot about how we were going to transition, and then we did it. And so this is a huge accomplishment, both for the Estuary Partnership as well as all the research groups who are involved in this and transitioning our 300-page EMP report into something that was much more manageable and it has multiple components. So we still have a classic PDF document report. This currently houses the executive summary, the background, the message. It has everything that the past EMP report had. But in those results sections, we have really pointed folks to go and view the interactive Tableau dashboards that host the data and allow you to interact and read those discussions and result summaries. Now, in that PDF of the report, which we're calling the hybrid report, we also have static images of the dashboards and the results. So that lives on in time, but we really would like people to engage in that online platform. Now, something that we're continuing to work on is to really minimize that PDF version of the report. We'd really like it to be a high-level analysis of what we see as stats and trends, and then point people to those Tableau dashboards for interacting with the data. And to do that, one of the things that we'd like to do is remove the method sections from the report. Right now, and historically, that ecosystem monitoring methods, all the researchers' methods, is almost a third of the document. It's a huge amount of information. And we wanted to pull that data out and put it into our protocol document. And so I'm going to just take a small sidetrack and talk about this new protocol document that we have published just this month, essentially. And let's go to the next slide and I'll talk about that. And this is an important part of this new hybrid approach to reporting. So this monitoring document is something that we started talking about in 2018 when we rolled out a supplemental information about how to best practices and technical details for monitoring water depth, water surface elevation, and temperature in the estuary. And back in 2018, we talked about how it would be really good to integrate best practices and technical details into the existing Roegner protocols. And this is a document, this is a SERP document that a lot of project sponsors and folks in the estuary use for informing how they monitor restoration projects through the Action Effectiveness Monitoring Program. And so we've been talking about this for a really long time. And then in 2019, December of 2019, we're like, you know what, we're rolling it out this year. We're gonna do a protocol update with a lot of technical details. We had a workshop where we talked to a bunch of people about their best practices, all the project sponsors. At that time, April Silva with Crest was also working in her best practices, working with Crest into her master's paper. And so we're like, all right, we're going to combine all these ideas. We're going to do a big update. COVID hit, as you know, and it's just been delayed. And the really good news about this delay is it has given us a lot of time to think about these protocols, this update, and make it so much more inclusive, including rolling in all of our researchers' methods into it. And so I know this is kind of a long story about it, but it's been a long journey, and we're just super excited to kind of get into the next steps. And how we see it now is more of a comprehensive guide to ecological monitoring in the estuary. It goes way beyond the scope of our action effectiveness monitoring and our ecosystem monitoring program. It includes methods that are applicable for really anyone who wants to jump into understanding research methods and monitoring in an estuary like the Columbia River estuary. And we have a really wonderful set of diverse authors, and it's really just an amazing resource. And so I can say a lot of really awesome things about it because I spent a lot of time reading and writing it with all of our co-authors, and I'm really excited to roll it out. So we have gotten some feedback in the last, in the last month since we've rolled out the new, what I'm calling version 1 of the protocols. And people they're like, well, what is this document? Is it fundamentally changing what we've already been doing? And the answer is no. The answer is that it is not fundamentally changing any of the methods that anyone has been applying in the estuary. What it is doing, it is taking those methods and it is providing much more detail for best practices when implementing those methods, as well as technical approaches that we find really useful for getting the most out of your method. And then in addition to sort of updating the existing methods that we see out of a lot of resources in the estuary and providing that technical expertise, it is also rolling in a lot of other methods, as I mentioned, both the EMP research methods and the action effectiveness monitoring, and to our water quality monitoring program that we run outside of the SERP. And so what are the next steps? Well, we really wanted to get version 1 done since we promised it in 2020, it didn't happen. So we have version 1 is now complete, and all of the EMP researchers have contributed. And now the next step is to essentially solicit feedback from our community. So we're going to be rolling that, we're going to send out an email, we're going to solicit feedback, and we're going to continue to update the document. If you read the document's introduction, it talks about this process and how we plan on updating it annually, and additionally continuing to keep these methods updated, not just in the document, but on monitoringmethods.org. All right, so that is my spiel, if you will, about the new protocol document and how it is an important component of this new hybrid report format, because we're going to be able to essentially get that EMP report down to something that is really bright and shiny and easy to consume, and then links to both the protocol document and our dashboards for folks who want more information. All right, so that is the plan. And now I'm going to return to our program of talking about the new report and the ecosystem monitoring different dashboards that we have. So this is the launching off space. And I don't know if Ian has shared a link for this yet, but we are going to share a link for the dashboards in the chat. And you can follow along if you would like as researchers talk about their dashboards and the data, and you can interact with it. This is our launch screen. So this is the essentially main dashboard where you can go in, you can read the executive summary, you can look at the background. It links to the PDF version of the report. It links to the protocols and other documents that you might find of interest. And it's really just a great place to really center yourself with the new dashboard system. We think that integrating the data across these Tableau dashboards for the EMP researchers is really going to help drive engagement with the data, not just for our EMP researchers themselves, but also for our community. And so we're really excited to hear how people are interacting with the data and what they might want to get out of it differently. And we'll have time to continue to have those conversations and evolve the program that we have. So what are our next steps? I hope I haven't talked too fast. I know I can get a little bit on a roll. I have to use my wizard hat to slow me down. We're really excited to continue, and you saw this slide last year, but we're really excited to continue this work towards integrating our researchers and our own data sets into this Tableau space. And the idea is, this year we did a really basic trends analysis with all of our researchers in that Tableau space, really trying to recreate some of the analysis they have year after year in the EMP report. If we can get that down, that's going to actually free the researchers up so they have more time for synthesis work, and they all want to ask more interesting questions utilizing each other's data. So we're going to continue to work towards integrating our Tableau dashboards towards a synthesis moment where folks can ask those big questions and utilize data across research groups. And of course, continue to update those protocol documents. So we're going to continue to work on that. If you would like more information about Tableau, because I didn't really dive into what Tableau is in this talk, because we actually did a really a good job last year, and I'm happy to share that presentation. But if you want to learn more about how to use Tableau, we are leading a workshop at SURF. And so all of my wizards are going to be leading that workshop. And if you are not available for the workshop or can't afford to attend it, don't worry. We are rolling out those materials after the workshop in a Tableau dashboard, and we can send it to you. All right, so without further ado, we can continue the rest of our presentations today. We have a lot of really good presentations coming up, and I'm so excited to have everybody here to talk about their journey and to talk about what they've seen in 2022 in the river and historically. If we have time today, we're going to have a great discussion on next steps and what folks in the community, the science work group community, think should be some of our next steps for the program. All right, so I will say that we will share this presentation. It has the links in it, and if you want to contact us with questions, feel free. I will stop sharing my screen.
So there is some time for questions. The next presentation's in a couple of minutes. So does anybody have any questions for Sarah so far?
I'm gonna put this up.
That was a lot.
I know. I did a good job keeping on time, so that means I probably talked too fast. So I apologize for that.
Hey, Kerry, do you have a question? Your hand is raised.
Yeah, it's a comment really quick. I was just realizing when you were talking, Sarah, just how much work you all have done. We've been focusing on our own dashboard and all our work, but you guys have done so much work. So that's awesome. Congrats.
Thank you, Kerry. It's been, this year has been a huge learning curve for all of us, and I'm really proud where we sit now, but I know that doing this transition has been a lot for everybody, so I'm really grateful. I'm really grateful for everybody's efforts.
Right, so one thing we could do, Sarah, is I could copy and paste a lot of the links and then send that out to the science work group so that they can have a lot of these Tableau links that they can go and check out later on after the meeting. Does that sound good?
Yeah.
Awesome. Any other questions so far? Alrighty, the next presentation is Joe Needoba, and he's going to talk about Mainstem and abiotic. Thanks, Lynn. And abiotic conditions. Joe, are you ready?
Yeah, hi. Okay, I share my screen. Can you hear me?
Can hear you. Oh, great. We can see your screen.
Okay, thank you, and thanks, Sarah, for nice introduction. Most of you have probably heard me give a similar talk each year about the water quality monitoring data that I've been doing as part of this project. I'm going to do that again today with some updates for 2023, but mostly focus on last year's data from our report and really emphasize our efforts to get this data available to everybody through Tableau. My name is Joe Needoba. I'm an associate professor, and I'm in the Oregon Health and Science University and Portland State University Joint School of Public Health. Within that entity, I teach environmental health to Masters of Public Health students and some graduate students, and we call our group is the Environmental Systems and Human Health group. So we're trying to relate the work we do to in one aspect to more of a public health, to get people engaged in the role of environmental science in public health. And that's the teaching side. And the research side, I'm very much involved in estuary biogeochemistry and how the things are changing in the estuary. So here is our landing page for the Mainstem and abiotic site conditions in Tableau. So one of the links Ian sent out will get you to here, and we have some a blurb about some of the results for 2022, and then you can look through some of the sections to get at the actual data that we've been collecting. What I'll say is that the transition has been challenging for sure. And I think this year we're going to see the usability and accessibility of this data to be much better as we get a handle on converting from our old file systems to these more dynamic data sets that are updated more often. We can really update them instead of once a year. We can just update them continuously as we get the data. And so there'll be hopefully more of a real time, or at least sooner access to data that we're collecting in the estuary. Sarah already said all this, so I won't go through it again, except that you can see the sites where we are collecting the in situ data that I'm going to talk about today, in situ water quality data. So Ilwaco, Welch, Whites, Campbell, and Franz. What I've been presenting is some of this is work that's been ongoing since 2008. That's when, as part of work we did with the National Science Foundation Center for Coastal Margin Observation and Prediction, we started putting instrumentation on at the time it was the USGS Beaver Army Terminal site, and it's shown here on the left, and that data was collected more or less from 2008 to 2018. We have periodically have some sensors on here now and hope to get funding in the future to continue that work with the USGS. And then on the right is the EMP program, where we have sensors deployed. You can see the pipe that is the PVC pipe there. The sensor is at the other end of that pipe underwater, but we use the pipe to securely house the sensors. And we're collecting things like temperature, conductivity, dissolved oxygen, pH, chlorophyll, as well as water samples to measure nutrients and the other parameters. So it says 16 years of data, but I'll show you some more granular resolution of that. I like to show these graphs from the Estuary Partnership. That the green here is existing priority habitat for juvenile salmon. The yellow is not currently habitat, but theoretically could be recovered, and the red is unrecoverable. So our sites in this reach of the estuary, Ilwaco and Welch, you can see where they exist in relation to some of the other yellow areas representative of this part of the estuary. And this is what they look like early in the season on the left at Ilwaco, later in the season in the middle on Welch. And just to show you that our pipes and where this data are collected are sort of how they look in relation to the habitat. We have to get to the Welch Island site on boat, and we've historically done it in a number of different ways, including going with NOAA and more recently with CREST. In this reach further inland, we have White's Island there in the green, and that site was also reached by boat, and you can see the pipe there almost completely underwater. We've extended that pipe all the way up to the shore so that we can get at it. So this one's over 30 feet long and allows us to continually collect good quality data despite low or high river conditions between March and September. In this reach of the river is Campbell Slough, and as many of you know, as you go further inland, the change in water level over the season becomes more significant, less tidal and more to do with the discharge from the watershed. And so on the left is a really high water year at Campbell Slough, where the pipe and all of the slough is basically underwater. In the middle is the opposite condition when water levels are really low, and you can see the trees are completely out of the water there. And so again, our approach with the pipe allows us to get both conditions and really get a good idea of the seasonality of this. And similarly at Franz, and you can see here that the difference at Franz is the mostly continuous presence of a beaver dam, and that presents some technical challenges to getting into where we have our sensor, but we're able to use our little boat to navigate that most of the year, although it's definitely a challenging site to continue to get in there at different water levels. So this is the sensor I want to talk about today. We're at this point, and next year, every single site will have a YSI EXO sensor, which we refurbish every winter, and then we maintain it for data quality purposes. We clean it at each time we visit, but it has a fairly extensive wiper system that keeps all of the sensors clean. And so you can see on the right, the business end of the water quality sonde has pH, chlorophyll, phycocyanin, fluorescence, which is a measure of cyanobacteria, as well as turbidity and dissolved oxygen. And the sensor also has a nitrate sensor, and so we've been experimenting with how well we can collect in situ nitrate concentrations from the sensor here. It is a little tricky. You have to correct for turbidity, but this year we're going to do an extensive analysis of how useful that sensor is. So here's our Tableau dashboard of the data that we've been collecting with the sondes over the years. And you can see when we started to work on this project around 2014, 2015 is when our lab began to service these sensors, the SONs, and we were able to incorporate these newer SONs from the old YSI models to the newer ones. And we were able to get a lot more coverage over the year, and it seems to be a lot better water. We often get better data because it's not, the biofouling isn't so severe, thanks to the new wiper systems. And so here's an example for temperature, dissolved oxygen, and chlorophyll. We have pretty good coverage, except for the COVID year. You can see we missed some data, unfortunately, but that was due to the shutdown and the inability to get out to those sites when we needed to. So I don't want to cover everything. I want to hit on a couple of things, and I just want to encourage people to reach out if they want to explore the data through Tableau. This is our commitment to the program this year, is to make that much more accessible, and we plan to do it. So Tableau does allow you to do some of these data analysis with the large data set a lot easier than if you were having to wrangle Excel files. Here's an output of the green bars here are temperature in the Mainstem, and is from a variety of different data sources, not just our EMP because it goes back to 1995. But these are the days of the number of days in a year where the Mainstem is over 19 degrees. And the red line shows the z-score, or you can think of it as the number of standard deviations from the mean. And so in 2011, we had a very relatively cold year, as in there weren't that many days where temperature went over 19 degrees in the Mainstem. And then we had a few years that are significantly warmer, especially 2015 and also 2021. Whereas 2022, the year we're reporting on at this meeting, was very much an average year for Mainstem temperature. Many of you have heard me talk about this before, but we really like to put the abiotic conditions in the context of river discharge, because it has such a big impact on especially on our EMP sites. That is like the size of the freshet, and and when it starts, as well as some of the spring or late winter rainfall events. And so all of these graphs are a little confusing at first, but the top line is the maximum river discharge of on a given day for any year, and the bottom line is the minimum, and then the purple line in the middle is the actual year. So in 2014, for example, you can see that river discharge followed very much the average, and we didn't have a lot of high or low numbers, whereas 2015 was a very low river discharge. There was hardly a freshet at all. And then 2017 was a very large freshet. And how these conditions affect our EMP sites is really the focus of our synthesis work in the coming year. More recent years are shown here, and in 2022. So you can see in 2021, I already mentioned that the Mainstem had a very high number of days where the temperature was above 19 degrees Celsius. You can see that the discharge in 2021 really followed the minimum we see for any year. And those two things are highly correlated, the temperature of the river and the amount of discharge. 2022, though, was a characterized by a big freshet, but not as long as what we saw in 2017. So these things I am actively incorporating into evaluating some of the water quality that we see. And I just want to try to tell that story today to a certain extent. So moving from the Mainstem to our sites, the takeaway message is that over the course of the months. This is again an output from Tableau showing a number of different, all of our sites, and showing the average temperature throughout the months, March through September. Across all the years, it's a consistent trend that Campbell and Franz have the highest number of days above 19 degrees Celsius. Ilwaco almost always has the lowest number of days, and that's due to the ocean influence at Ilwaco, where colder water is often present. And Whites and Welch are very much representative of the Mainstem in terms of they're much more connected to the Mainstem, and so they often look a lot like what the Mainstem temperatures are. Another way to visualize that data is here throughout the years. And so in this sense, we can see Campbell and Franz 2014 and 2015 had a lot of warm temperature days, and again in 2021. So this type of visualization gives you a really good overview without, you know, in sort of simplifies the way to look at some of this data. 2022 then was primarily less warm days, but you can see that that compared to Ilwaco, Welch, and Whites, Campbell and Franz still showed some of those warm days. So while summer temperatures can exceed 19 degrees at all the sites, Ilwaco really has the coolest at the longest periods, and the hottest temperatures are seen at Campbell and Franz Lake. Tanya's going to talk about chlorophyll a bit later today. I just wanted to point out that in addition, that Campbell and Franz stand out from our grab samples to have relatively higher chlorophyll, which is a representation of the algae, the microscopic algae that are growing in these sites. And we often see very high chlorophyll in Campbell and Franz that exceed 30 micrograms per liter, which would be considered eutrophic or very high productivity areas. And that is the context to interpret the dissolved oxygen data as well as the chlorophyll data that we're collecting with the SONS. And so here's our the in situ chlorophyll data from Campbell, Franz, and Welch. And just looking at this graph, you can kind of get the sense that there's an early spring growth period where chlorophyll is high at all the sites, and it's highest at Campbell, which is the blue line. And then chlorophyll drops down, and this is always the result of the freshet moving through, which really knocks down the productivity of algae. You know, there's still obviously growth happening in the marsh plants. And then the summer period is characterized by these sort of dynamic changes, the booms and busts of chlorophyll. So river phytoplankton show seasonal changes with highest densities prior to the freshet. In the summer, though, some of the chlorophyll is associated with cyanobacteria that can grow to very high densities and detected by our pigment fluorescence phycocyanin, which is also on those sensors. And we're starting to look at the dynamics of when and how cyanobacteria tend to take over at these sites. So then I want to spend a couple of minutes on dissolved oxygen. And this is another way that Tableau you can process the data. This is showing all of the sites, the average of all of the data. And so you kind of get the sense that from this, that dissolved oxygen as a water quality metric, it looks pretty good. If you look at the top graph, now that's the average of all the sites of all the years, and they all sort of, you know, are similar in the spring, around 100% saturation, meaning that they're more or less in equilibrium with the atmosphere, and that there's some changes in the fall, and that has to do with the productivity of the sites. But really, the average is not a good indicator of what's going on in these systems. You have to look at the higher temporal resolution data, and we collect this data every 30 minutes, and so that's what's shown in the bottom graph here. And so if you just focus on the blue line, that was Campbell Slough early in February, March timeframe. It's the data does hover around 100% saturation, but as growth begins, we see both very high levels of oxygen on a given day and very low levels. And so that swing represents productivity in the daytime, respiration at night, and in some cases, when conditions are right, we see oxygen going right to zero in Campbell Slough and in Ilwaco in particular. And so in 2022, which I'm showing in the bottom graph here now, in particular, we saw this event at Campbell Slough where oxygen went to zero for over a week and then gradually recovered again. And I've looked at this extensively and ruled out biofouling as a problem. And I just want to talk about how that dynamic change happened in Campbell Slough. So falling back on the trusty Excel graphs, I can demonstrate this a little better. I talked about this at our CREC meeting this year as well, and had some good discussions with people there. What I'm showing here in black is the water depth. So the freshet can be interpreted. You can see where the freshet occurs in May and early June with a peak in June. Dissolved oxygen, this is the average over the course of a given day plotted over the time frame. And so this is a daily average dissolved oxygen in percent saturation. And the dynamic that we see is that the freshet really peaks. And if you imagine those pictures I showed before of Campbell Slough, the water is at a very high level. The pipe is basically underwater and the entire field is underwater. And then as it recedes, we see this significant drop in dissolved oxygen down to zero, and that lasts for several days. And it really corresponded with this idea that if you look at the bottom graph of chlorophyll and phycocyanin, that there's very little chlorophyll at the peak of the freshet, but just as it begins to recede, there's a large change in fluorescence, presumably a lot of growth in the water that leads to a decrease in dissolved oxygen as the water recedes. And I can just only imagine that there's a lot of respiration happening at this time following this large growth period. And then it recovers for the rest of the summer, where we don't see any more, despite a lot of primary production and presumably a lot of aerobic respiration, the slough never goes back to these low oxygen conditions. And this is a pattern that we see year after year, but this is the first time we've seen oxygen go this low. But 2023, this year, we saw the same thing, but not to quite the extent. And so the black line is the depth from 2023. You can see the freshet kind of had a peak again in late May. There was a little bit more dynamics to the discharge in this case, but dissolved oxygen was high in the spring, sort of reflecting on productivity. And then as the water receded after the freshet, we saw a large dip in dissolved oxygen, and then it recovered again. So 2 years in a row, we see this repeating pattern. Lowest dissolved oxygen following the peak of the freshet. The chlorophyll was captured really well in 2023, and I have noticed this trend before, but it was really obvious in this data that the water levels in Campbell Slough going up and down on almost a spring neat tidal cycle pattern. Every time the water recedes, we see a big increase in chlorophyll. And so there's a relationship between whether water levels are increasing, which seems to not promote phytoplankton growth, to then water levels are decreasing and we get these big peaks. And so this is something that we're really going to try to quantify rather than just showing the trends to understand better how tightly correlated these changes are. I wanted to explore that a little more by looking at some of the other years. I know I'm running out of time, but 2017 again had one of the largest fresh hits. You can see on the left there that Campbell Slough, everything is underwater. And again, if we look at chlorophyll and depth, there's a phytoplankton bloom early in the spring following a decrease in water levels. And then again in July, when water levels finally decreased after that large fresh hit, we'd see another large bloom of chlorophyll. That also corresponded with phycocyanin. So in addition to chlorophyll, there's definitely cyanobacteria growing there. And again, those trends in dissolved oxygen tend to show a significant decrease every time the water level is decreasing versus increasing. So I hadn't in the past really honed in on that particular pattern, but given that Campbell Slough is essentially the most disconnected from the main stem, it really responds to these changes in water level that are still ultimately driven by the discharge. So I'd say that dissolved oxygen levels decrease in shallow wetland habitats, but it's modulated by water depth. During the peak flow, there's less opportunity for convective exchange, and dissolved oxygen saturation can decline. And I think that's partly what's going on. And so that as the water stagnates, as it's beginning to recede, dissolved oxygen gets drawn down by respiration. I look to pursue that hypothesis a little more with our data analysis this year. And so that's the take-home message, is that at Campbell Slough, I just thought I'd focus on that today. Water levels, you can stand there and it's only up to your knees at some times, and then at other times it's 15 feet over where that person is standing. So really dynamic system, and means a lot for water quality. So this year, off-channel wetland sites have revealed that there's clearly conditions that are good and also sometimes poor. Low dissolved oxygen is evident in some of these sites every year, and I'm beginning to understand the drivers of that low oxygen. Warmer water, algal blooms, and hypoxia develop as a result of these interrelated effects, and timing of the spring freshet could be a mechanism for understanding this habitat quality. So I just want to point out, so it's myself and Tawnya Peterson that are leading a lot of this effort. We have this year the help of two great students, Allison Meister and Jordan Lancaster. That's Allison there up in Franz Slough, and we managed to get the boat over the beaver dam and get our samples. And so this is challenging work to get these samples, and so I really appreciate the support, and in particular to get to some of our sites. Narayan and April have been really awesome this year, and just wanted to thank them. So with that, I could take some questions. That's my presentation.
Great, thanks, Joe. We have time for like one question, and then I think we're going to have time at the end of the meeting if there's any other questions. Jason.
Hi. Yes, great talk, Joe, and thanks for letting me ask this question now, because I'm an ecologist trying to ask an abiotic question, which takes my brain a moment. So Joe, I was just wondering if with all these years of sampling you've done, and the patterns you're seeing, do you think you would be able to be predictive at this point for future, like say for 2024, if you saw something in March with timing of the freshet or something going on that you would say, hey, everyone, look out for July, because I think we'll see this pattern in other water quality measurements like DO. And if that's the case, if there'd be like any management implications for that, that could be acted on kind of in real time.
I think so. I think that those patterns I was trying to explain today are repeated enough time. We've have enough examples of very different conditions. I mean, 2011 was a good year. We don't have very good data from 2011 to show what it's like when there's an extremely large snowpack that leads to a really big freshet. But 2017 and at least 2017 is a really good example. And so I used to think that a large freshet ought to mean like, you know, really good habitat quality because there's so much water. But the receding of that large freshet in all of these years has been followed by large cyanobacteria blooms and low dissolved oxygen. And so starting there, I think we can make some predictive, you know, we can predict what it might be like, and we can definitely maybe alter our sampling to better capture that. I mean, the sensors are out there, which is great. But we could plan when we go out there a little more specifically to try to capture, try to take samples and see what's really going on. So my hope is to turn this discussion into a really quantitative assessment of these trends with the idea that if we can see that it's that 2024 is going to have a really low snowpack, maybe it's an El Nino this year and it's too warm in the winter time to have a large snowpack at the end of the spring, we should be able to predict what's going to happen based on the last few years. Essentially, I'm kind of saying that it's not so simple that low discharge leads to poor water quality in the estuary. It does certainly impact temperature, but it seems to be the opposite, that the large discharge leads to these really big changes in chlorophyll and dissolved oxygen.
Great, thanks. Joe, I have a couple questions, but I will hold them until later. So I'm gonna turn it over to Sarah and Sneha. You're gonna talk about habitat structure.
Yes, Sneha's taking the charge. Her wizardiness is gonna just do it. Go, Sneha, go.
Yeah, I'm just gonna share my screen real quick. And hopefully it continues. Oh, there it goes.
Yep, got it. Thanks.
And I'm still sharing the presentation screen, right?
Yeah,
I always have like a problem with like the screens because now they have this new presentation mode and I'm like, where did the screen share go? Anyway, thank you, Sarah and Joe, for those awesome presentations. I will be talking about the habitat structure dashboard that we released as part of the 2023 EMP hybrid report. Okay, so we at LCEP, with the support of CREST, monitor the following habitat components. We evaluate the interannual variation in indentation pattern across 6 trend sites. And based on these indentation patterns, we also study the variability in vegetation, sediment dynamics, as well as soil composition at these sites. In addition to these, we also collect above ground plant matter and detritus samples at a subset of our EMP sites, which help us to evaluate the quantity and quality of biomass, which feeds into the larger food web study of the ecosystem monitoring program. This is the 2023 map of the sites that we sampled at. We also monitor habitat conditions at Cunningham Lake, and we collect biomass samples at Steamboat Slough. I started doing this last year, where I talk about like the number of plots and everything that we collect, like the basically to show the scale at which we collect the data. And this year, I want to focus everyone's attention on the amount of drone data that we have collected over the summer. It's a royal we, because I was off for the summer taking care of my little baby, while Sarah and Ian and the rest of the field crew got a new drone, which is LiDAR enabled and has a huge multispectral sensor. And they collected a total of 8 terabytes of drone data, which Ian has been really great at processing some of this data. And at the end of the presentation, I can show you like a quick look of our initial images that we were able to create. Sarah touched a lot about the report that we released this year. So I'm just going to say that this year, the report has 2 sections. One is a written report, and the other one is a collection of Tableau dashboards. The written report has an executive summary. It has background information about the program, as well as some of the static results and discussions. The report also has links to the Tableau dashboards that we and our research partners have created. The Tableau dashboards are standalone dashboards that have interconnectivity, which makes it very easy for the user to toggle between different research focuses. Here is the habitat structure dashboard that we at LCEP created. This is the introduction page. And on this page, you can see an overview map of the sites. Adjacent to the map, you can see these navigation buttons, which will allow you to go into the different sections in the dashboard. We also have an executive summary for the 2022 trends. Below the map, you can see navigation buttons that will allow you to go to dashboards that were created by our other research partners. And below this, there is a link at the bottom of the page to the written report of the 2023 EMP hybrid report. Here in the corner, there is also like a link if you all want to access the habitat structure dashboard later on after the presentations are done. As we moved all of our data into Tableau, we were also able to create this giant inventory of the data that we've collected over the past several years, like starting from 2005. The program has evolved, and we've been able to show the evolution of the data collected. And as you can see, the sites have basically data available from like 2008. Soil chemistry was introduced into the program in 2017, and all the way at the bottom of the page, you can see the evolution of monitoring protocol that we went through to be able to collect good biomass data. Moving on to the results, or like the ecological component dashboards. The first one that I will be talking about is the hydrology dashboard. We have at the very top of the dashboards, and this is something that's common across all of the dashboards. We start off with an overview map, and that map essentially allows you to change the visualizations of graphs on your screen. I have defaulted this to Welch Island, but in case you want to see multiple sites at the same time, you can always control-click the sites of interest. Each of these sites have a hydrograph, a temperature graph, percent exceedance graph, and a habitat opportunity analysis. The percent exceedance graph, we have overlaid that with the site elevation histogram, and this allows us to show different indentation patterns on the site. And this in turn helps us evaluate or have like a thought process of how how the wedge community and the sediment is going to move around in the site. We have also added filters for the different sampling years in case you all want to look at a certain site, how it's behaving, for example, in 2011 when it was a really high fresh rate, or 2015 when there was a really low fresh rate. Next, we move on to the sediment accretion dashboard. All of these dashboards you can access when you go, when it's right there next to the overview map. Again, the sediment accretion dashboard shows you the results for net accretion rate and the net change in sediment accretion across our EMP sites. If you want to take a look at multiple sites, again, you can always control-click. Another analysis that we were able to incorporate into this study after we moved our data to Tableau was understanding how our wetlands are keeping track with sea level rise. For this model, we used the Army Corps' Columbia River Adaptive Hydraulics model, and it basically provides sea level rise in meters across 50 years, 75 years, and 100 years. When we compared the net accretion and erosion rates of the EMP sites to these scenarios, we saw that currently Franz Lake is the only site that is keeping up with these scenarios. I have to admit that these scenarios are pretty aggressive as well. Now we'll move on to the soil development dashboards. We have collected data since 2017, and we have data available for 2017, 2018, and 2021 and 2022. We have, since this is a new monitoring aspect, we have taken some time to hone in on the protocol to make sure that the data quality is good. Similar to the previous two dashboards, right at the top of the page, you have an overview map. Adjacent to the map, you have these black bars that will allow you to navigate. We also have, so each of these buttons on the map will allow you to change the data visualizations. We also have a link to a poster that Derek presented at the CREC conference, and I have added that to the slide here. The reason I'm showing you this is because I wanted to talk about how we were able to create analysis by integrating Tableau into our workflow. And we were able to formulate and analyze some hypotheses. And we discovered that as an overall trend, reintroducing tidal flooding changes the soil profile to restore and leads changes to microbial activities to help in successful native plant community establishment. It also helps us to study the soil composition that can lead to decreased prevalence of reed canary grass in restoration sites. Next, we move on to the plant community development dashboard. This dashboard gives us results for plant cover and proportion of native versus non-native plants across the EMP sites. At the very bottom of the page, we were also able to show the interannual variability of plant diversity across our EMP sites. And what we noticed was that Welch Island consistently had high diversity. The other thing we noticed was based on the freshet, there were dips in species diversity that we saw in the more fluvial reaches of the site. The reason we study habitats or plant community not only gives us information about habitat structure, but it also is an indication for primary productivity, which we have analyzed in the biomass analysis dashboard. This dashboard, in addition to showing the changes in biomass quantity as living dead and species. Also shows the chemical composition of dominant species at the site. And what I mean by that is we were able to send the samples for analysis, and we got back results for carbon, nitrogen, which basically are an indicator for the amount of energy that the plants can provide to the food web. It also gives us a quantitative measurement for lignin, which is talks about the structural components of the plant. And basically, it means that if there is a lot of lignin in a plant, it's going to take a long time to decompose. In addition to this, we also have information about the inorganic nutrients present in this present in the plants, and this is a measure of like disease resistance, etc., in the plants. What we noticed is that among the 5 dominant species on our trend sites, reed canary grass took a long time to decompose, and native species like wapato and lingvii sedge took less time to decompose because they had more carbon and nitrogen content. A simple analogy for this is if you think of a charcuterie board, reed canary grass is like the celery that no one wants, and it's always there it's left over in the board. Similarly, in the winter times, reed canary grass is predominantly left on the site. And if y'all are wondering why I'm thinking of charcuterie boards, it's because I just noticed it and I like it, and I'm a millennial, so I love my charcuterie. Well, these are all the dashboards that we have released this year. And while, as Sarah said, that these are just like the first iterations, and we'll obviously, iron out the kinks and release more synthesis later on. However, by just the first iteration, we have been able to release some online publications. The first one was the Walewski study, where we combined the wedge data with the UAV data, and we have a lot of other manuscripts in prep. As we continue to hone in on our data sets, we can provide a more complete story of the habitat structure of our EMP sites. And this will allow us to also improve and introduce new data sets. And an example of what that could look like is on this slide. This is, so Ian was very fast in grabbing the LiDAR data off of the Walewski flight that they conducted this fall. That's basically like literally just a month ago. And he processed it to show a true color image, an elevation classification, as well as a habitat classification. On this slide, I've also mentioned like the accuracy, the vertical and horizontal accuracy. And moving forward, we aim to introduce these kinds of products into our workflows. This effort we don't do this work in a silo. We work with April and Ryan from Crest. We have an amazing field crew in our office, as well as Jody Reed and Kim Biafra from SA. And we're grateful to everyone who comes out to the field and enjoys with us, like bearing my jokes and everything. So thank you very much, everyone. And at this point, I will take any questions if anyone has them.
Great, thanks, Sneha. Any questions? I have one really quick. Are you planning on doing or is Ian going to do the drone imagery that you just showed in that last slide with all of the ecosystem monitoring sites as well?
That's the plan.
Perfect. Awesome. Okay. Derek looks really happy in that photo back there. So
processing biomass samples is the best.
He looks like he's having fun. Any questions for Sneha? That was a really, really good presentation.
Thanks.
That might have been like hour 5 of washing those off.
No questions? Ready. We'll have another time for more questions at the end of the meeting. So the last presentation before the break is Tawnya Peterson, and you're going to talk about conditions affecting juvenile salmon food web.
Yeah, thank you. Let's see. I don't use Teams that much for sharing my screen. Oh gosh. Should have looked at this before. Oh, shit, there it is. Never mind. And I apologize. I have to go right after I finish because I'm teaching right after this downtown. So I need to do that. Screen share.
Yep. Got it.
Okay.
Yep.
Perfect.
Perfect. Thank you.
And okay. Awesome. Well, what a great presentation, Sneha, Joe, Sarah. It's such a lot of work, and it's just, I think Sarah already said it, it's really kind of an honor and a privilege to be part of a group that's so committed to doing this kind of work, and it's, you know, it's icing on the cake to have great people to work with. So I've always been excited. I continue to be excited, and every year that we collect more data, I'm even more excited to be part of this group. So thank you all. So I'll talk a little bit about some of the work that we do, sort of the opposite approach to Sneha. I'm not going to cover all of it. I'm going to just kind of hone in on a couple of different things. You are welcome to visit the dashboard and see some of the other data that we're collecting. And I will kind of touch on it, but not really go into a lot of detail. So I am Tawnya. I'm, along with Joe, part of the OHSU PSU School of Public Health. I'm the program director for the MPH program in environmental systems and human health. So I like to sort of see the human dimensions in all the work that we do, and the students really help with that. We've got a couple of great students, as Joe mentioned, Jordan and Allison, who are working with us right now, and then students who've contributed to the work that went into the last fiscal year's worth of data, Kelsey and David. They did some great work in the lab and in the field. And just by way of introduction, Sarah requested that we do a little bit of that. Our data sets include nutrients, chlorophyll, plankton species composition and abundance, and then natural abundance, stable isotopes of carbon and nitrogen in a variety of substrates. We look at dissolved nitrate to try to understand sources and patterns of sort of the inorganic part of the food web that feeds into the primary production and goes up the chain to the fish. And then we collect information about primary producers, salmon prey, and then salmon tissues for the purpose of trying to estimate what the salmon are eating in the food web. So we do some food web modeling using those isotopes. Okay, there. Our dashboard looks like this. This is about the last time I'll show this, but when you look at it, you will see there are a variety of different sections you can navigate to that tell you more information about each of the components of what we do in the plankton ecology section. And I always like to show this because this encapsulates what we're trying to get at. The EMP is trying to figure out what the habitat quality is looking like for salmon. Is it suitable? Is it not suitable? And our approach is to look at what's at the bottom, so the primary producers, either plants or phytoplankton, that feed organic carbon into invertebrates that then get eaten by the fish. And we're really trying to get a better understanding of what the habitat looks like for fish, and that all starts with the base of the food web and what they do to influence water quality. A lot of that's driven by phytoplankton, as Sneha was mentioning, microbial processes as well. So we can get some hints about what that looks like from some of the analysis that we do. All right, so I'm going to talk about a few little stories in the data that we have. I don't know if I'll get through all of them actually, because I feel like I have too many slides, but I'll do the best that I can. Joe touched on this already, but the influence of river flow variation on primary production is a theme that we've seen for many years, and I think we're getting a better handle on the specifics of that and what it looks like. And I think if this is one area that I think we could do a really nice job synthesizing. So Sneha just referenced the influence of river discharge on plant communities and some of the the species composition and productivity. I think that's an area we could come together and get some good synthesis and analysis. I'll talk a little bit about the climatology of chlorophyll and nutrients. Because we have many years of data, we can do this now, and it's really interesting and exciting to be able to show quantities and kind of impart some predictability, kind of to Jason's point earlier. I will talk a little bit about the variation in carbon and nitrogen isotope signatures, and this helps us understand a little bit about what's available at the base of the food web to drive ecosystem productivity up to the fish, and then a little bit about the nutrients themselves. So discharge patterns, we are all pretty familiar, and thank you for the introductions earlier already, so this should be pretty familiar. The top plot here is showing the discharge pattern colored by, this is chlorophyll on the top, and the different colors are oxygen saturation. So you can sort of do this kind of thing where you look in the winter, there's low oxygen saturation because primary production is low. And during these peaks, you get higher oxygen production from the phytoplankton. And this coincides with the river discharge pattern. So we kind of know that this is true. So the chlorophyll peaks, algal biomass peaks in the spring when there's enough light availability and nutrients. And as soon as the freshet comes in, it kind of knocks it down. So you'll see this peak happens just before this red line, which is the discharge for that year. That knocks down the quantity of algal biomass in the water, and you tend to see a decline. And as Joe was talking about, this has some implications for kind of who wins in the aftermath, and we can pay attention to what that looks like and make some predictions. So it's not surprising that there's a relationship between primary production and river discharge, because you get the effect of dilution, and that is somewhat, we understand it. But it's important to emphasize that the when is really important, particularly when you're talking about biological quantities like plants and invertebrates. The when is really important, and the timing of when the phytoplankton are increasing in abundance and who they are influences what happens with the zooplankton and so on. So one example that I sort of noticed in exploring the data this year in Tableau, and this is a Tableau plot, was there was an interesting relationship between the amount of water held behind the Grand Coulee Dam. And so you see here on the right, here's an image of the Columbia River Basin. And way up here in this box is Grand Coulee Dam, and Lake Roosevelt lies behind it. And below here is a picture of what that looks like. And the amount of water held behind there is important because you get growth of phytoplankton that's not diluted by river flow. It's just kind of hanging out there. And at some point, when that water is released in advance of the freshet, you get that slug of water making its way downstream. And so that's what this image is showing. This elevation declines somewhere between April, May, June, depending on the year. So I have a number of years here, 2018 through 2022. And I just chose one of them, probably not the best one, 2021, where you can see, oh, actually, I'm sorry, this shaded area is offset here, so ignore that for now. But the dip in water elevation means that water is going downstream in the river. And here's a plot of the chlorophyll from that same time series at 3 of our EMP sites, Campbell, Franz, and Welch. And around that same time period, there's a peak in chlorophyll at Franz and Welch, and a decline or a dip at Campbell. And so the timing is pretty aligned, making it, we can hypothesize that there is that slug of water moving down that increases chlorophyll at some of the sites, and it appears to decrease at Campbell, and that's only because the chlorophyll was increasing to a level that was higher than that lake. And so what we see is a dilution of that effect. So you can see that depending on the site, depending on what the river flow is doing, there might be different things happening. And sort of, I guess, to follow on from Joe's presentation, those patterns at the off-channel sites like Campbell, which are quite separated from the Mainstem, you can see you can imagine there being predictability there to say if that chlorophyll is increasing and you dilute it out, you might get a period of decreased algal abundance, and that might maybe protect water quality for a little while, only to then have it change later. So there's things we can predict from these relationships. Moving on to here's another Tableau plot showing climatology of nutrients at the various EMP sites from Campbell, Franz, Ilwaco, Welch, and Whites. I haven't figured out how to not do this alphabetically in Tableau, but here it is. Usually I like to do it from reach A up to reach F, but we are alphabetical here. And the purpose in showing this, so each site is shown here with a time series over the year. And what you can see from these climatologies is that there are certain periods of time during the year when year-to-year variability is highest. And that's important to know because then we can dig into those time periods and try to figure out more specific relationships within them. So for nitrate, the greatest variability is seen in that winter-spring transition time. So you see the widest variability in these box plots early in the year. And that's different for phosphate. And so those two nutrients contribute to primary production. And so we can understand a little bit about what drives phytoplankton productivity during those time periods based on the nutrient availability. So you can also see where which sites tend to have the highest nutrient concentrations. And that would be sort of Campbell and Franz and Welch and Whites have pretty much the same in the winter. And then later on in the year, you can see some sites differing. And then an interesting thing to note about the phosphate is that Franz tends to have, often has quite high phosphate later in the year. And I think this is potentially important in driving cyanobacteria blooms at that site, but we need to figure out a little bit more about where that phosphate's coming from. Another view of the nutrients looks like this. This is the time series on the x-axis showing just time, and this is year-to-year variability. Each of these peaks in the time series is roughly the same, which tells us that each year nitrate concentrations in the beginning of the year, in winter, spring, early spring, are quite similar. So if we saw something different, we might make some predictions about how that translated to biological productivity in the system for that year. So they're pretty much all hovering around 40 at Franz and Campbell, a little bit higher at El Waco. That's a very different site. So each year seems to look pretty similar in the winter. This plot, I don't expect anyone to really look at and digest in any amount of time. But what I've done here is I've plotted all of the nitrate data and then divided it into clusters based on concentration. So this is the whole thing you can look at. Don't worry about it too much. What I'll show you next is how those clusters turned out. So we can see that if you divide the clusters of nitrate into different concentrations from low to high, you can see the number of samples we have in each of these concentrations. And you get a sense that it's very rare to have very high concentrations of nitrate. It's only 3 samples in in our entire data set that have concentrations in the 50s. And then most of our samples are taken are occurring when the nitrate concentrations are about 4. So this gives us an idea of what the normal is. And I think that's it's very powerful in a data set to know what is normal and what is higher or lower than expected, because that tells us when something rare occurs, and we can kind of get a handle on how rare it is. So generally speaking, the late winter, early spring, nitrate concentrations are somewhere between 20 and 25, or I guess probably this cluster 3, 30. And then you get the rare occasion where it's higher or lower. The low ones reflect drawdown and growth by phytoplankton. And we can also get a sense for how extreme that might be. We can look at the monthly climatology of chlorophyll, and Joe already talked about this. Franz stands out here as having a really high value of chlorophyll in 2017, like an excessively high value. It's a very strange observation, but that was during that period of very high river flow. But the values at Franz are always more extreme than other places. And this kind of goes along with just the type of site that it is. The beaver dam is present, and so it cuts off circulation for certain times of the year. But you can get a sense here of, what's normal? What kind of variability would we expect? And so we have very low values, hovering around 0, up to sometimes in the 20s, 30s, 40s. And I will say that the water quality standards suggest that observations 3 months in a row of grab samples that are greater than 15 micrograms per liter are considered impaired. And so we don't generally have that, but there are times when we're getting some of our observations exceeding that. It doesn't last that long, but we should pay attention to what's happening there. And I will say I feel like those standards are a little bit antiquated in the sense that we have hourly data we can look at to say, like, how long did it last for? And, a grab sample doesn't always tell you the whole story. This is just to show you what 2022 looked like. If you're interested in this year's data, there was nothing exceeding about 10 micrograms per liter. So we were in good shape in 2022. All right, so one of the questions we were interested in knowing, and not just us, but lots of estuarine and river-dominated estuarine environments, are off-channel habitats more biologically productive? Do they contribute subsidies to a river ecosystem? And there's some suggestion that this is the case. We see high chlorophyll values in some of these off-channel habitats, and we see a larger dynamic ranges of dissolved oxygen and changes in pH, which suggests that there's a lot of activity there. And whether that makes its way into the river and how much that contributes to the food web are things we're trying to figure out. And so we can use nitrogen isotope data to get to provide more insight into that as well. So we have our quantities that we're measuring on a consistent basis, plus we have these other alternate strategies to learn some things. And so if we look at, I'll just to briefly remind folks, stable isotopes are, they occur naturally, and a very small fraction of the nitrogen and carbon out there is so-called heavy. So carbon is usually has a mass of 12. There's a small fraction of the amount of carbon out there that has 13. So you can use that small fraction relative to the more typical fraction to understand or to kind of trace different sources of carbon, and similarly for nitrogen through the system. And we can measure this and by assuming that the isotope ratio of the sample minus the standard, which is just the standard that everybody uses, and we report it in units of per mil, and then we can employ Bayesian mixing models to figure out what sources of prey contribute to the salmon food web. And I'm not going to talk about that today, but it will be in the Tableau report when I'm still working through linking our code into the Tableau interface, but I'm getting there. Okay, so just looking at the isotope ratios of, Sarah, do you have a question? I saw a hand raised, I think. Did I?
Sorry, I just clicked on the wrong button. Keep
Okay.
going, you're doing great.
All right, so we use the isotope ratios of the particulate organic matter, which is the stuff that basically builds the whole food web. We can learn some things that might be important for our food web modeling. So number one, we make assumptions in food web models that the isotope signatures at the base of the food web are kind of constant. And that's not necessarily totally true. And it's particularly in a dynamic system like a huge river, it's good to know how far we can push that assumption, and also what things we can learn from it. So what I'm showing here is a plot showing our particulate organic carbon and in different size fractions over the course of a year. And so July through, this is several years worth of data, 2009 through 12. And the black bars are the total amount of POC, and the gray is that's contained within large diatoms that are phytoplankton that tend to be very nutritious for salmon prey and then into salmon. And so what I'm showing here is a sequence of observations that go along with this time series that shows the signature of carbon and nitrogen through the course of this bloom is actually changing. And so in the early phase of the bloom, leading up to the peak, the carbon and nitrogen are similar to each other. And then after the bloom, they change. And this is kind of speaking to what Joe was saying earlier about that sort of the receding part of the bloom. You might see a change in the species composition that might have a slightly different signature, and the abundance, which would then, the species composition is likely reflected by the carbon, and the nitrogen could be that plus utilization of nitrogen from the inorganic source. Point being, there are differences you can see, and when you have observations in your total number of samples that are quite close together, any information you can have to disentangle those is really helpful. So by interpreting these values that we get relative to the ecological situation, we can better understand what we're dealing with for the food web modeling. So this is really important to try to understand the processes, like what's leading to differences in those signatures. And then we can use that to better understand what the salmon are eating later down the road. So getting to the last bit, we can use the inorganic nitrate. So this is the inorganic nutrient source that the primary producers use to build biomass. And then that biomass gets eaten by the prey and then the salmon. And those, we assume that the signatures in that inorganic source are going to be the same, and then we use that assumption in our food web modeling. But again, that's not necessarily true. And we can use the isotopes of nitrogen and oxygen together to try to understand something about what sources of nitrate are coming into the system and how that might be making its way into the food web. So this plot is just showing an initial analysis showing that the signature of nitrogen in nitrate over time is it increases over time, which is logical because when the phytoplankton are taking up the nitrogen from nitrate, they're leaving behind the lighter source, N14 dominated source, and taking up the heavier. So that when you have a heavy signature left behind, it means this phytoplankton are taking up the nitrate and leaving behind, which goes along with our earlier analyses of the timing of nutrient drawdown. So early in the season, lots of nitrate that gets drawn down and taken up by the phytoplankton, and the nitrate isotopes are agreeing with this. And this is important again for the feeding into the food web modeling to get a better and more precise idea of what the prey sources look like. And then to get back to our question about whether the off-channel sites are more productive than the main stem, I grouped, so these are the nitrate isotopes, the nitrogen isotopes of nitrate in the various sites, Campbell, Franz, Welch, Whites, and then an average of all the off-channel compared to what we have for the main stem. And the off-channel ones are heavier, which implies potentially that that uptake of nitrogen in nitrate is higher in those off-channel sites, leaving behind the heavier source. There are other things that potentially are going on, and we can explore this with a dual isotope approach. But at first pass, that is supporting evidence that those off-channel sites have greater ecological productivity. To sort of just point out some of the things that we'll be getting to next over the next little while. This is a biplot of the isotopes of nitrogen in nitrate compared to oxygen in nitrate. And you can use this information compared to some known quantities that are shown on the left to pick out different sources of nitrate, which is important to understand as it's fueling the biomass in algae in the river. The one that stands out that's kind of cool is this one where there's an isotope of nitrogen and nitrate that's near zero and quite high in the oxygen. And this is characteristic of nitrate fertilizer. So we can pick out sources like that by using this dual isotope approach. That might be helpful in interpreting some of the patterns that we see. Finally, here's another example of the benefit of a dual isotope approach, where we have data from Ilwaco in Reach A, which is organized by the abundance or the signature of nitrogen and nitrate from low to high on the top. And then the same exact sites are showing oxygen. And what you can see is that the negative values of oxygen are associated with marine intrusions that occur at Ilwaco. You would never know that from the nitrogen, but you can see it in the oxygen. So that helps us to disentangle some of the sources that we might see in nutrients throughout the system. So next steps in our world are to work through the species composition and determination of phytoplankton and zooplankton, as well as the abundance, up to the end of 2023, with the help of mainly of Jordan, helping to process the prey and POM samples for carbon and nitrogen isotope analysis, as well as the N and O of nitrate to try to get at these sources. Nutrients need to be run, and I'm gonna get better at Tableau. I'm gonna do it, and then continue to work on the Bayesian mixing model to feed in more data. Getting the data back from the isotopes can take quite a while, so we're kind of a little bit, we're a few months behind in terms of collecting and getting that data analyzed, but we're going to be getting some soon. And I hope to work with the folks at UW on comparing what we know about the stomach contents in the prey with what the food web models can tell us. because they match up pretty well, and it would be good to be able to feed that into the models more precisely. And then I want to say one quick thing about, two things. One thing here is that we've been collecting the carbon and nitrogen isotopes of reed canary grass. It's always there, so it's consistent. We can get samples of every time we're out. And what this is showing is the average carbon isotope signature in reed canary grass changes over the course of the year, which I think can help us. If we see detrital samples in the river, for example, we can compare that back to the time that it might have been produced, and it might help us to sort of understand the contribution of that source to detrital pools. So it's kind of interesting. You can actually see a difference. It's not the same value all the time. Again, sort of reinforcing this idea that we feed this data into a food web model, but we need to understand that it's not always the same. And then as we work through the information, kind of thinking about the uncertainties that we could address, the ERTIC uncertainties, our time series of data, now that it's getting long enough to be able to think about climate change effects and ecosystem, the response to the ecosystem, which might inform strategies for restoration and mitigating negative effects, trying to understand sources of nutrients and productivity patterns. And then thinking about water management and